Hi, everyone. Thanks for coming today. I'm really excited to introduce Dr. Piyali Sengupta. So Piyali did her postdoc work with Corey Bargman, where she was the first person to identify and clone an olfactory receptor in C. elegans. And since then, her lab has investigated a wide variety of sensory behaviors. For some reason, I always think of thermosensation, but she's done more than that. And I think what really stands out to me is she's combined these molecular and circuit approaches to understanding the sensory nervous system, which is something that you frankly just don't see in other animals very much. So I'm really excited to hear about her work today on the interaction between the gut and the nervous system. So welcome, Piyali.
All right, let's see.
Great.
Is that working?
Yep.
Okay. So, okay. Well, thanks, Adam, for inviting me to share our work at this virtual forum. I've actually very much enjoyed listening to talks here over the last few months, and I'm actually very glad to hear that you're going to continue this through the rest of the year. And I also wanted to thank all of you for tuning in wherever in the world you are. So I don't typically do this in normal seminars, but these are not normal times. And so I thought I'd start out with a couple of very brief comments. So I'm an immigrant scientist. I grew up in Kolkata, India, and I came to the US at the age of 18 for my undergraduate work. And I've stayed in this country ever since, moved around different places for graduate and postdoc work. As you all know, all this kind of moving around isn't particularly unusual for scientists. We tend to try and go wherever we can go to do the work that we really want to do. And over my career, I've had the great privilege and honor of mentoring students and postdocs who have come from all over the world. And like many of you, I also have collaborators in many different countries as well. So I'm now a naturalized US citizen, but over the last couple of years, 3.5 to be exact, it's become increasingly challenging to see the rise of xenophobia and racism that is preventing people from getting the same opportunities that I was provided when I first came to the US and holding back scientific and intellectual progress. So while it may be simplest to actually ignore everything and just do your science, I think it's become even more important now for us to speak up, push back against these artificial borders and barriers and against this blatant disregard for science. So I think we must all do what we can to try and restore some semblance of sanity and humanity to this world. So here's hoping for a new and improved 2021. Okay, so moving on to the science. So my lab actually works in a few different areas of sensory biology, but the overall question that my lab is interested in is understanding how animals sense the world around us and how they translate this information into specific changes in behavior, but also development. So as you're all aware, a really characteristic feature of behavior is that it must be both robust as well as flexible. So behaviors must be robust because animals need to be able to execute these behaviors over a very wide range of conditions. But conversely, behaviors must also be flexible because animals need to change their behaviors adaptively, depending upon what's going on around them, and what their past experience has been, and what their internal conditions are. So today what I'm going to do is tell you about how internal state modifies neuron and circuit properties to result in sensory behavioral plasticity. So let me first start out by defining what I mean by internal state. So I'm defining internal state as a global state that is established as a consequence of an animal's physiological conditions and also experience. So internal states such as hunger, reproductive state, sleep, play a very important role in determining how an animal is going to respond to an incoming sensory stimulus. So for example, a mouse, so I realize this is an invertebrate forum, so this is the only vertebrate example I'm going to show. So a female mouse will respond completely differently to odors or pheromones rather present in the urine of male mice, depending upon what part of the estrous cycle she's in. And similarly, she will also respond completely differently to pup odors, depending upon her sexual experience and also her hormonal conditions. And so work done in many labs, Richard Axel, Catherine Dulac, Lisa Stowers, Kazu Tuhara, Neuro Shah, and others, have shown that internal state in this case modifies circuit properties at many different levels to result in changes in behavior. So under some conditions, the responses of sensory neurons in the periphery themselves are altered, but more commonly, information processing is altered in central brain regions to result in these behavioral plasticity. So a very well studied and well understood internal state, of course, is that of hunger. So hunger is a major behavioral drive. And as you're all aware, it essentially will completely reprioritize and override even your most well-intentioned decisions. And so hunger will greatly affect how an animal is going to respond to an incoming sensory cue. So for instance, a hungry fly will respond with much more strongly to attractive chemicals and will decrease its responses to chemicals, to repellents, than as compared to a fed fly. And so work in many labs, Jing Wang, Kristen Scott, David Anderson, and others have shown that internal satiety state in this case is read out by neuropeptidergic and hormonal signaling to both modify responses of gustatory and olfactory neurons in the periphery, but also chemosensory information processing in brain regions such as in the mushroom bodies to result in these observed altered behaviors. So, but how does this kind of integration and modulation occur in a nervous system that is not only structurally but also numerically far simpler? So C. elegans the C. elegans hermaphrodite nervous system has about 300 neurons, and of these, 60 of them are sensory, and so these are indicated here by these triangles. So compare that to millions of olfactory neurons, just olfactory neurons in a mouse, and a few thousand olfactory and gustatory neurons in flies. And so these neurons are also organized quite differently. So the majority of axonal processes are present in this structure called the nerve ring, with the cell bodies present in these ganglia in the head, but also organized along this ventral nerve cord. So despite this relative simplicity of the nervous system, C. elegans nevertheless exhibits extremely complex and highly experienced dependent sensory behaviors. So in the lab, we grow C. elegans on a plate of bacteria. But in the wild, C. elegans actually lives in a much more complex environment. And so there's very nice work done now on the natural ecology of C. elegans. And this is work by Marianne Felix and others. And so they've shown that in the wild, you typically find C. elegans associated with microbe-rich rotting organic matter, like rotting fruits and stems, like you would find in an orchard, for instance. And so under these conditions, worms need to be able to respond both robustly as well as flexibly to many different sensory cues, including light, chemicals, touch, and temperature for optimal survival and reproduction. So just like in any other animal, a major driver of how a worm is going to respond to sensory cues is whether it's well-fed or whether it's hungry. So as I mentioned, we grow C. elegans in the lab on a plate of bacteria, and typically we give it a very boring diet of a single bacterial E. coli strain called OP50. But in the wild, it has a much more varied diet, and it associates with and consumes an entire smorgasbord of different bacterial strains. And so some of these bacteria are just food, some are pathogens and parasites, and much less well-studied are bacteria that exist in a commensal and mutualistic relationship with the worm. So bacteria such as OP50 typically are not alive in the worm gut. So there's a structure in the pharynx called a grinder. And as you can imagine from the name, it grinds up any bacteria that's sort of going through it. And so here, this OP50 strain has been transfected with a red reporter. And you can see that it's, you can see this sort of red diffuse fluorescence in the gut, and there are no colonies. But a lot of the other bacterial strains that worms will eat in the wild will actually survive and colonize the worm gut. So for example, this strain called Providencia JUB39, you can see these colonies that are present in the posterior part of the gut. And in this beautiful picture from Buck Samuel's lab, you can see in red these bacteria that are present in the worm gut that survive and colonize the intestine. And so these types of bacteria that actually are alive in the gut, so they have the potential or the capacity to not only just provide nutrition, but also to modulate worm physiology and behavior via the production of bacterial metabolites. So I'm going to tell you two stories today. So the first one is an unpublished story on how the complete absence of bacterial food, or starvation, reprioritizes the worm's responses to temperature cues in its environment. And in the second story, I'm going to tell you, it's a recently published work on how a gut colonizing commensal bacteria modulates the olfactory behavior of worms via the production of a neurotransmitter. So the first story is the work primarily of two postdocs in the lab, Asuka Takeishi, who a few months ago left to start her own lab at the Riken Center for Brain Science in Japan, and Jamie Yun, and with a lot of contributions also from a third postdoc in the lab, Nathan Harris. So like chemosensation, thermosensation is also an incredibly important sensory modality in C. elegans. So as I said, C. elegans is associated with the soil, and the soil is subject to very large temperature fluctuations in the course of a day. So in this simulation, you can see the temperature changes in the soil as a function of time of the day and depth from the soil surface. So C. elegans is an ectotherm. It does not have internal thermoregulation. And so the only way it can actually maintain a constant body temperature within its physiological temperature range under these conditions is by altering its behavior. And as it turns out, worms actually exhibit extremely complex, highly experience-dependent thermosensory behaviors. So this was first described by Ed Hedgecock and Dick Russell back in 1975. So without getting into too much detail, essentially, if you take worms don't essentially have a preferred set temperature. So if you take worms and put them on a thermal gradient, the temperature they're going to seek out on that gradient really depends upon the temperature that they were exposed to by 3 to 4 hours prior to the assay. So for example, if you grow worms at 15 degrees and you put them on the gradient at 20 degrees, they're actually going to seek out cooler temperatures. And so this is a behavior called negative thermotaxis. And conversely, if you grow worms at 25 degrees and put them at 20 degrees, they go in the opposite direction. They move towards the warmer part of the gradient in a behavior called positive thermotaxis. And so we've actually done a lot of work on understanding how worms change their behaviors in an experience-dependent way, how they respond to small temperature changes in the environment. But that's a topic of a completely different talk. And so all I'm going to talk about today is how the presence and absence of bacterial food actually modulates these behaviors. So what I'm showing you here is the behavior of very well-fed, happy worms, or presumably happy worms. But it's also been known, and Hedgecock and Russell also showed this, that if you deprive worms of bacterial food, then thermotaxis behavior is completely disrupted. So in this assay performed by Nathan, you can see that if the worms have been starved for about a couple of hours, then essentially both the negative as well as positive thermotaxis behavior are disrupted. And this is a little hard to see, but what they actually end up doing is they make a lot of tight reversals and turns at the starting temperature. And so this ends up that the worms actually have a prolonged residence at the starting temperature and don't migrate up or down the gradient. So this has been known for 45 years, and with the exception, however, of a single very nice paper from Ikwe Mori's lab in between, essentially how satiety state modulates thermotaxis behaviors has remained a mystery. So positive thermotaxis is somewhat more finicky, so we haven't actually looked at that in a lot more detail. So all I'm going to talk about is how starvation disrupts negative thermotaxis behavior. So directed navigation behavior, such as negative thermotaxis, is mediated via modulation of the worm's underlying locomotor behavioral strategy. So if you take a worm and put it on a solid substrate such as agar, it's going to go off in a given direction. I'm going to call that, or it's called a forward run. But randomly, it'll stop, reverse, turn, and then head off in a completely different direction. And so negative thermotaxis is actually mediated in part via a locomotory behavioral strategy called biased random walk or clinokinesis. And so this was first described for thermotaxis by Will Ryu and Aravi Samuel, and also by John Pierce and Sean Lockery. And so the idea is pretty straightforward. So when it's moving in the preferred direction, which when it's performing negative thermotaxis would be in the cold direction, it essentially suppresses the execution of reversals and turns. And so run duration is increased. Conversely, when it's moving in the warmer direction, which is the non-preferred direction, it increases reversals and turns, and therefore runs are terminated. And the net result of this, of course, is net migration towards the colder part of the plate. And so we calculate a metric that we call the thermotaxis bias, which is essentially run duration towards the cold minus run duration towards the warm over total run duration. And somewhat confusingly, a positive number means negative thermotaxis. So as I said, well-fed animals perform nice, robust negative thermotaxis, and you have to starve them for about 2 to 3 hours for this disruption of negative thermotaxis. And essentially what this means is that now they're no longer modulating their churning frequency as a function of temperature changes on the gradient. So clinokinesis or biased random walk is one of the strategies. They also actually use another strategy for effective negative thermotaxis. So when they make a turn, as I said, then following the turn, the run is again headed in a different direction. And what has been shown again by Aravi Samuel's lab is that a fed animal performing negative thermotaxis, their run direction following a turn is preferentially biased towards the colder part of the plate. But what Asuka also found was that if you starve these animals, then essentially the run direction following turn is pretty much randomized. And one thing to note here is that they're no longer actually biasing their runs either towards the cold or the warm, and it's essentially pretty much random on the gradient. And so this also takes 2 to 3 hours to manifest. But both of these behavioral strategies, however, are completely reversible. And so if you refeed the animals for about an hour with bacteria, negative thermotaxis is restored. And so what that also tells us is that starvation does not result in kind of irreversible changes in the underlying circuit. So one thing to note here, I'm not going to show you the data for this, is that the worms really do have to eat the bacteria. You can feed them either live or antibiotic-killed bacteria, but they have to eat it. So if you just expose them to the odors of the bacteria, that is not sufficient to mimic the fed condition. So another question is, well, how is it that starvation, how does it end up disrupting negative thermotaxis? So the major thermosensory neurons in C. elegans is a pair of these neurons in the head of the animals called AFD. There's a left and a right. And their major postsynaptic partners are these interneurons called AIY, again, a left and a right. So I told you that in both mice as well as in flies, one of the ways that satiety state will modulate sensory behaviors is by actually altering or tuning responses in the sensory neurons themselves. And so the simplest hypothesis for us was that starvation essentially disrupts thermosensation in the AFD neurons. And so Asuka took a look at this. And so you're going to be seeing a few of these through this part of my talk. So this is going to be a rising temperature stimulus, and the extent of the temperature range is shown here on the left. And in these heat maps, each of these lines is the response of a single neuron, in this case expressing GCAM. And so what you can see here is that in response to this rising temperature stimulus, AFD neurons respond very robustly with an increase in calcium, intracellular calcium. They start responding when the temperature reaches a certain threshold. And without getting into this in too much detail, that threshold happens to be set by the acclimation temperature of the worm. So this is in fed animals, and what happens upon starvation, and pretty much nothing. So one thing you'll probably notice here is that there is a small change in the amplitude, but in other experiments we know that this level of change in the response amplitude is not sufficient to disrupt negative thermotaxis. So if temperature responses in AFD are not being modulated by starvation, the other simple possibility was that starvation might actually be altering the synaptic output of AFD, so either facilitating or disrupting synaptic output. And so this can be assessed by looking at temperature responses in the AIY interneurons. And so Asuka also looked at this. And so here again is a rising temperature stimulus. And here's the responses of individual AIY interneurons organized by the initial time of response time. So one thing you will already notice is that these interneurons have a very different way of responding to temperature. So this is true for a lot of interneuron responses to different sensory stimuli. They have a more stochastic response. But overall, what we can see is that the response probability or the total response duration or even the average response duration per neuron is not significantly altered by starvation. So one thing we do notice, as you're probably also noticing, is that there are more neurons that do start responding at a lower temperature, but what the behavioral significance of this is unclear. So essentially, I think it appears that upon starvation, the responses of the core thermosensory circuit, which is the AFD and AIY neurons, is pretty much unaltered. So how else is satiety state information brought into the circuit? So a few years ago, both my lab and Ikue Mori's lab had identified another neuron, another sensory neuron, that also responded to temperature, but it does so in a very different way that I'll show you in a minute. But so these are the AWC neurons. And at least under the conditions in which we do our experiments, if we actually silence these neurons or physically remove them from the circuit by ablations, we actually didn't see much of an effect on thermotaxis behaviors. And so exactly what it was doing in the context of thermosensation was really unclear. So AWC is actually not known as a thermosensory neuron, but it's extensively studied as an olfactory neuron. And this is work from Corey Bargman, Shrikant Chalasani, Noel Latoile, and others. So AWC responds to bacterially derived odors. And it has also been shown by a number of labs that the responses to these odors can actually be modified by the satiety state of the worm. And so this seemed like a really good candidate to be bringing in, integrating feeding state information into the thermosensory circuit. And so Suka decided to take a look at this. So I told you that AWC responds to temperature. And so she wanted to know if under starved conditions, if the temperature responses would be modified. And just to remind you, the AFD temperature responses were not significantly altered upon starvation. So here are the responses of AWC neurons, and you can immediately see that it's very different from the responses in AFD, and it's actually fairly unusual in terms of the way most sensory neurons in C. elegans respond to stimuli. And I can discuss this at the very end if anyone's interested, or speculate on it if anyone's interested. But essentially what we see is these stochastic responses, but both the response probability as well as the response duration per neuron is actually significantly increased upon starvation. So if this temperature induced activity in AWC is increased upon starvation, and if that actually does play a role in disruption of negative thermotaxis, one prediction that we would make is that if we could silence these neurons while the animals are performing thermotaxis, perhaps that would be sufficient to restore negative thermotaxis. And so Jamie tried this experiment, and she did this using optogenetics, using this light-gated anion channel. And you can see here that when these animals in the presence of retinol and light, as the animals are performing thermotaxis, if she silences just the AWC neurons, that is sufficient to fully restore negative thermotaxis. And essentially the animals look like they're not starved at all. So one thing here to point out, which I will also mention at sort of near the end of this section, is that what this also tells us is that the functions of the core circuit are really not altered, but essentially their output is being masked by the activity state of the AWC neuron. So how is increased temperature responses in AWC result in disruption of negative thermotaxis? So this is the sort of the small part of the circuit in C. elegans, and I'm just showing you the AFD and AWC neurons and their major postsynaptic partners. And given the small size of the nervous system in worms, it's not particularly surprising that they actually share many of these primary or first layer interneurons in common. So one thing I already mentioned was that the way worms perform negative thermotaxis is via modulation of turning frequency. So when they're moving down towards the colder part of the plate, they really have to suppress turns and reversals so they can continue to move towards the cold part. And so work primarily from Corey's lab has previously shown that if you activate AWC, either optogenetically or via odors, then the worms this actually promotes reversals. And so one idea we had was that if you actually, in starved worms, have increased temperature-induced activity, perhaps that actually results in increased turning and disrupts AFD-mediated negative thermotaxis. And in fact, in a recent paper earlier this year from Ikuemori's lab, where they were imaging freely moving animals, they actually showed a decorrelation between temperature responses in AFD and turn frequency as the worms are performing thermotaxis on these gradients. So how would increased temperature responses in AWC be read out through the circuit to actually result in changes in reversals and turns? So this is actually pretty well studied. So AWC inhibits the AIY and AIA first primary interneurons and activates the AIB interneurons. And so we wanted to know whether in starved animals, the increased temperature responses in AWC, how that's read out by the circuit to result in altered behavior. So we looked at temperature-induced calcium flux in all these first interneurons. And so I'm not going to show you all the data for this, but what we found is, so AIY I already showed you, there's no changes in temperature responses upon starvation. And somewhat surprisingly, AIB interneurons also we saw no changes. But in AIA, there was a pretty significant change. And just to remind you, AWC inhibits AIA. And so the idea would be that if you see increased temperature responses in AWC, you should be seeing actually decreased temperature responses in AIA in starved animals. And so Suka found this to be the case. So here what she finds is that the AIA interneurons actually show these tonic temperature responses in fed animals in response to a rising temperature stimulus, but these responses are quite significantly suppressed upon starvation. So if this reduced temperature responses in AIA, again, are causal to the disruption of negative thermotaxis, you might also predict that if you could activate these neurons while the animals are performing thermotaxis, you could perhaps restore negative thermotaxis behavior. And so Jamie did this experiment, and she activated them using optogenetics again. And you can see if the AIA interneurons are activated as the animals are navigating the thermal gradient, that is sufficient to restore negative thermotaxis even in these starved animals. So now the question was, well, so how is it that the internal state is being read out by AWC as well as AIA to actually alter their temperature responses? So one major readout of satiety state, of course, is insulin signaling. So this is true in humans, it's true in mice, true in flies, and also true in worms. So for example, in flies, it was shown by Jing Wang many years ago, Jing Wang's lab, that in starved flies, low insulin signaling modulates presynaptic facilitation at this first sensory synapse, and this actually promotes food-finding behaviors by hungry flies. So the C. elegans genome actually encodes 40 insulin-like peptides, but luckily for us, we didn't actually have to go searching through all 40 of them. We had a very good idea of which insulin-like peptide was going to be involved, and this is the Insulin-1 insulin gene. And we knew this was going to be involved, or we had a very good guess this was going to be involved, because it's already been implicated in starvation-dependent plasticity in salt responses and odor responses by both Yuichi Ino's lab and Cory Bargman's lab. But more importantly, Ikue's lab had previously already identified this gene or this ligand in mediating starvation-dependent plasticity and thermotaxis. So several years ago, they had actually done a forward genetic screen looking for mutants where animals no longer disrupted negative thermotaxis upon starvation, and that isolated an allele of INS1. And so consistent with what they had reported, indeed, we find that in INS1 null mutants, the animals essentially act like they're, at least in the context of thermotaxis, they essentially act like they're fed even when they're starved, and they perform negative thermotaxis just fine. So we wanted to know, and this has been a little bit unclear, what the required source of insulin signaling is. And so these experiments have actually previously been done via overexpression of the insulin ligand in various tissues and Insulin-1 null mutant background. But we wanted to sort of avoid the caveats that are associated with the overexpression of a diffusible ligand. And so what we wanted to do is knock out the insulin gene specifically in different cells and tissues, and then try and see which tissue was actually the source for this ligand. So insulin-1 is actually expressed in a bunch of neurons in the head, but also very strongly in the gut. And so to knock it out in these different tissues, Jamie used a Krelox-mediated recombination. And I'm just going to show you a subset of her data. And so what she found was that knocking it out it is actually expressed in AIA, but knocking it out there had no effect. But if she knocked it out in the gut, that completely restored negative thermotaxis behavior, just like to the same extent as the INS1 null mutant, suggesting that the intestine potentially is the source of insulin ligand in starved animals. So if the gut is producing insulin, and so we wanted to know whether ultimately it actually targets the AWC neurons to modulate the temperature responses and thermotaxis behavior. So the readout of insulin signaling in mouse contexts is the DAF16 phosphotranscription factor. And Ikue's lab, as well in the context of thermotaxis, but also in several other contexts, it's been shown that Insulin-1 actually antagonizes DAF2 insulin receptor-mediated signaling. And so the idea is that if insulin is antagonizing the insulin signaling pathway, this would result in dephosphorylation of the DAF16 phosphotranscription factor, nuclear translocation, and then presumably changes in gene expression. So what this would also predict then is if AWC happens to be the target of insulin signaling, if you could actually knock out DAF16 specifically in AWC, that should actually mimic the Insulin-1 mutant phenotype. So in order to do this, what Jamie did is she took advantage of this auxin-induced protein degradation system that was implemented in C. elegans by Abby Dernberg's lab. And so the idea here is that we have the DAF16 gene that is tagged with this degron sequence by gene editing, so it's done endogenously. You express this F-box protein called TIR1, which in the presence of auxin essentially targets this degron tag protein to the proteasome, and then it undergoes degradation. And so what this allows us to do is to provide both spatial and temporal control of protein depletion. So when she did this experiment, she knocked out DAF16, specifically in AWC, upon addition of auxin. And what she found was that addition of auxin during the starvation was actually sufficient to, again, completely restore negative thermotaxis behavior, suggesting that DAF16 indeed acts in the AWC neurons to modulate this or disrupt negative thermotaxis. So now we had this question of, so if insulin signaling from the gut is either directly or indirectly targeting AWC via DAF16, but in insulin 1 mutants, the negative thermotaxis is completely fine even upon starvation, what happens to AWC temperature responses in starved animals? According to the insulin 1 mutant phenotype, the prediction would be that AWC neurons should actually be in insulin 1 mutants be completely indifferent to feeding state. And it should respond similarly both in fed as well as in starved animals. And moreover, their temperature responses should resemble those in fed animals. And so Suka looked at this. And so what you're looking at here in the dashed lines are the temperature responses in AWC in wild type animals. So black in this case is fed. And as I showed you before, temperature responses are increased upon starvation in wild type. But you can very clearly see here that in insulin 1 mutants, however, there is no difference in the temperature responses between fed and starved in AWC. And also more importantly, the responses resemble those in fed wild-type animals. And this is particularly marked in the AIA interneurons because here, as I showed you before, again, the dashed lines are wild-type responses. The responses are significantly suppressed upon starvation. But again, in these solid lines, you can see that pretty much the temperature responses in AIA are again completely indifferent to feeding state and resemble those in the AIA interneurons in fed animals. And so putting this together, so the current model here is that the presence of bacterial food in the gut inhibits insulin signaling. And under these conditions, the AWC neurons are fairly quiet and AIA is active. And what we think is that this temperature responses in AIA, this activity, is actually permissive for AFT-mediated modulation of turning frequency and the execution of negative thermotaxis. However, in the absence of bacterial food, insulin signaling from the gut, either directly or indirectly, antagonizes this pathway in AWC. This results in increased temperature responses in AWC, decreased temperature responses in AIA, and this in turn results in increased turning and decorrelated turning, resulting in disruption of negative thermotaxis behavior. So we think this is a pretty nice example of a situation where the functions of the core thermosensory circuit, their functionality really isn't specifically altered, but their output is actually being masked by the activity state of a modulatory circuit who's as a function of both satiety and gut-brain signaling in this case. And there's a preprint on this if anyone is interested in reading about this further. So what might be the advantage of such a mechanism? So this is a bit of a speculation, but one of the things we have also found previously is that upon starvation, the AWC actually starts responding much more strongly to chemical odors, so potentially bacterial food-related odors. And so one speculation here is that this particular mechanism simultaneously allows AWC to increase its responses to food-derived odors, while at the same time deprioritizing thermoregulatory behavioral strategies. This remains to be actually directly proven. So the known unknowns, so we have no idea how insulin signaling is being regulated by starvation. It does not appear to be regulated at the level of transcription in the gut, so there might be other mechanisms in play. We don't know whether there are additional insulins that are intermediate or whether insulin 1 directly targets AWC. And currently, we have no idea what the targets of DAF16 are in AWC. And so these are some important experiments that are currently ongoing, partly in my lab, but a lot of this also in Asuka's own lab at Riken. Okay, so far I've talked about bacteria essentially just as a food source. And that is really true for OP50, which, as I mentioned before, typically is not alive in the worm gut. But I also told you that there are bacterial strains that worms will eat in the wild, which are happily survive and colonize the worm gut. And so in the second part of my talk, I'm going to tell you about how bacteria can be more than just a food source. So this is all the work of a postdoc in the lab, Mike O'Donnell. Actually, he's a postdoc for about 2 more days, and then he is leaving to start his own lab at Yale. And also a really wonderful past research associate, Henry Chow. And this has been an ongoing, really wonderful collaboration with 2 chemists, Bennett Fox and Frank Schroeder. So Mike came to my lab with a very specific interest in understanding interorganismal interactions. And he was particularly interested in seeing whether bacteria that exist in a commensal relationship with C. elegans, whether they actually modulate behavior in any way. So he messed around with a whole bunch of different bacteria and a bunch of behaviors. And so he settled on this particular bacteria called Providencia JUB39, which is a mouthful, so I'm just going to refer to it as JU39 from now on. And he showed that this bacteria colonizes the worm gut. And I should mention here, this is a highly nutritious, non-pathogenic bacteria. And he also tried out different behaviors. And so what he found was that colonization by this bacteria, gut colonization by this bacteria, results in altered responses of the worm to a bacterially derived odorant called Octanol. So Octanol is this long-chain alcohol produced by a bunch of different bacteria, but at very high concentrations, worms actually really dislike it and they will avoid it. And you can assay this by using this incredibly high-tech assay. You essentially take one of your eyelash hairs or eyebrow hairs, you glue it on a stick. So the assay is called smell on a stick. You glue it on a stick, you dip it in Octanol and stick that in front of a worm that's moving forward. And essentially, you quantify the response latency for it to move backwards. So the longer it takes to reverse, the less well it avoids this particular chemical. And so what Mike found was that OP50-fed animals have respond or back up really quickly. So this has been described before. But interestingly, he found that JU39-fed animals, however, take a much longer time to reverse, showing that their aversion to this chemical is significantly decreased. And so going forward, I'm going to show you the reversal latency in the form of these bar graphs, where each of these dots is going to be the relative response latency of a single animal. And so a positive number is going to indicate reduced aversion. And I'm not going to show you the data for this. So as I said, this work has just recently been published. Mike went on to confirm that you actually need live JU39 in the gut. So dead bacteria do not result in this modulation. And the extent of aversion, essentially, or the extent of a reduction of aversion is correlated with the extent of colonization by this bacteria, gut colonization by this bacteria. So what could the mechanism be by which colonization by JU39 was resulting in reduced octanol aversion? So octanol is sensed by a single pair of sensory neurons in the head of the worm. It's a nociceptive sensory neurons called the ASH neurons. And so work done primarily by Rick Komanuki's lab and others have shown that the extent of octanol aversion is actually modulated by a number of monoamines. And so one of the hypotheses that we had was that perhaps what JU39 was doing was targeting one of these monoamine systems and thereby reducing octanol aversion. So there's no effect on dopamine and serotonin, and so we focused on octopamine and tyramine, which is the invertebrate homologs of the adrenergic system. And the biosynthetic pathway for the production of these neurotransmitters is known, and this was worked out by Mark Alkema. And so worms take up tyrosine. This is then converted to tyramine by this enzyme called tyrosine decarboxylase. And tyramine is then converted to octopamine by this enzyme called tyramine beta hydroxylase. And so mutations in TBH1, there is no octopamine that's produced. Mutations in TDC1 take out both tyramine as well as octopamine. And so any of the phenotypes of TBH1 are typically also shared by TDC1 mutants because in both cases there's no octopamine. So to see if this pathway was actually targeted by JU39, what Mike did was he took TDC1 or TBH1 mutants, grew them on OP50 or JU39, and then quantified their response latency to octanol. So as I showed you before, wild-type animals fed on JU39 showed this reduced octanol aversion. And what he found was that in TBH1 mutants, that was abolished. And so that told us that octopamine signaling in the worm was important. He then looked at TDC1 mutants, and what he found was TDC1 mutants essentially look like wild-type animals. And so this was a bit of a surprise because, as I said, phenotypes of TBH1 mutants are typically shared by TDC1 mutants. So one difference between TBH1 and TDC1, of course, is that in TBH1 mutants, you should expect a buildup of tyramine. And so we thought, well, maybe that was one of the reasons. But then when he looked at TDC1, TBH1 double mutants, again, they look like TBH1 mutants alone. So this actually threw us for a loop for quite a while. And then Mike came up with this hypothesis that he could explain all of this if he could propose that Ju39 itself produced tyramine. And then the worms took this tyramine and converted it to octopamine by their own TBH1 enzyme. And so the idea here would be that Ju39 produced tyramine would compensate or would complement essentially the absence of tyramine production by worms in TDC1 mutants. And this would pretty much totally explain the phenotypes that he was seeing. So to see if this hypothesis had any basis in reality, what he did was he took wild type and TDC1 mutants, grew them on OP50 and Ju39, and then we sent these cultures to Bennett and Frank at Cornell. And so they performed HPLC and high-resolution mass spectroscopy to look at the levels of tyramine. So have wild-type worms grown on OP50 or Ju39, both have plenty of tyramine. TDC1 mutants grown on OP50 do have none, and this was already shown before by Mark Alkema. But sort of gratifyingly, what they found was that if you take TDC1 mutant worms, which remember are not making their own tyramine, and grow them on Ju39, there was plenty of tyramine restored. And one interesting thing here that I can discuss at the very end if anyone's interested is that if you just take the bacterial cultures alone, you actually don't detect tyramine. The bacteria have to be actually present along with the worms. So how do these bacteria actually make tyramine? So the fact that bacteria make tyramine is actually not particularly novel. So this has been known for over 100 years. And so there are these gram-positive bacteria like Enterococcus and Lactobacillus that make tyramine, and it's actually present in foods like aged cheeses and cured meats. And so these bacteria have this operon in their genome where there's a tyrosine transporter and this tyrosine decarboxylase enzyme that converts tyrosine to tyramine. So tyramine production has not, as far as we knew, had not been reported in gram-negative bacteria, which is what Ju39 is. But undeterred, Mike went ahead and sequenced the genome of Providencia. And what he found was that there were two enzymes encoded by the genome, which by their sequence and structure looked like they had the potential to actually be tyrosine decarboxylase enzymes. And so to see if they were actually required for the production of tyramine, he then knocked them out. He grew worms on these mutant bacteria, and again, we sent the cultures to Bennett and Frank. And so here's a TDC1 mutant worm. Again, this mutant worm is not producing any tyramine of its own. On OP50, there's no tyramine. On Ju39, there's plenty of tyramine. If you grow them on these mutant bacteria, you can see tyramine production is completely abolished. But more tellingly, if you actually just look overall at the abundance of different metabolites whose abundance is changed upon mutating these two genes in this bacteria, all of these metabolites that are affected are primarily tyramine-derived metabolites. And that's actually important because that also establishes that mutating these two genes in this bacteria doesn't result in global changes in either bacterial or worm metabolism. So one key question here, of course, is that if you now grow worms on these mutant bacteria, what happens to octanol aversion? And so Mike did that experiment. And so what you can see here is that if you grow worms on these mutant bacteria that do not make tyramine, they also do not show that reduced octanol aversion that we typically observe when they're grown on the wild type Ju39. So, and I'm not going to show you data for this, but it's in the paper, but he also confirmed that this mutant bacteria can colonize the worm gut just fine. So the model that we're building up then is that the worms eat the bacteria that colonizes the worm gut. This then produces tyramine. The worm converts it to octopamine, which then modulates octanol aversion. So this type of a model would also predict that if you actually provided a source of exogenous tyramine to the worm, that should also be taken up by the worm, converted to octopamine, and should also reduce octanol aversion. And moreover, exogenous tyramine should actually be sufficient to complement the absence of tyramine production by these mutant Ju39 bacteria. And so Mike also looked at this. And so here he's growing worms on OP50. And so what he finds is that just on OP50, and remember, OP50 doesn't make tyramine. If he adds exogenous tyramine to the growth conditions, you can show reduced octanol aversion. Addition of tyramine does not further reduce octanol aversion in Ju39, but it is sufficient to again reduce octanol aversion if you're growing these worms on these mutant Ju39 that do not make tyramine on their own. Okay, so far, we have the worm taking bacterially made tyramine, converting it to octopamine. And so now the question was, where is this octopamine acting in the worm's, the host's nervous system? So the worm genome encodes several octopamine receptors, which are actually expressed fairly broadly in the nervous system. But remember, I told you that Octanol is sensed by the ASH sensory neurons, and so we wanted to know whether that itself was the direct target. So there are 3 octopamine receptors expressed by the ASH neurons, and so Mike looked at these. So mutations in SER3 or TYRA2 have no effect, but mutations on OCTR-1, however, completely wiped out the reduced Octanol aversion, and this could again be restored by putting back wild-type OCTR-1 sequences specifically in the ASH neurons, suggesting that octopamine is directly targeting the ASH neurons. So at this point, so we have this whole pathway going from a colonizing gut bacteria, production of a neurotransmitter, conversion to a different neurotransmitter by the host, and then targeting the host nervous system to result in this modulation of an olfactory behavior. So at this point, you might be wondering, as we were, what's the point of all of this? Why does the bacteria go to such lengths to actually modulate octanol aversion? So as I mentioned before, octanol is actually a bacterially derived odorant. So we do not know whether JU39 itself makes octanol, but there are other bacteria that do. And work from Liz Glader's lab had previously shown that JU39 actually does make a whole bunch of other types of volatile odorants, including alcohols that at high concentrations, like octanol, are also very aversive to the worm. And so we thought that perhaps one of the reasons this was happening is these bacteria, by altering the chemosensory behaviors of the worm, in one extreme was manipulating the feeding decisions of the animal and promoting or biasing the worm to choose that specific bacteria for consumption. And so this could actually have benefits for both the worm and the bacteria. It could benefit the worm because, as I mentioned, bacteria like JU39 is highly nutritious, and could also benefit the bacteria because it could allow colonization, stable association, and dispersal. And so Mike developed this food choice assay where he grew worms on different types of bacteria and then gave them a choice between 2 lawns of either OP50 or JU39, and then looked to see which bacteria the worms chose. And what he found was that worms fed on JU39 preferentially chose JU39 for consumption. But if the worms were actually fed the JU39 that cannot make tyramine, then they no longer chose JU39. And I'm not going to show you the data for lack of time, but this also requires that the worms actually make their own octopamine, and also requires that OCTR-1 receptor as well. So this idea of microbes in the gut actually modulating or specifying sort of dietary decisions of the host has been described before. So there's some really nice work from Carlos Ribera's lab, where they showed that if you deprive flies of amino acids, then they preferentially choose protein-rich foods such as yeast for consumption. But if you have a specific combination of bacteria in their guts, then they actually no longer choose these yeast, even though they are deprived of amino acids, although all the underlying mechanisms are not completely clear. And so coming back to the major theme of my talk, so this provides, I think, a nice example, we think, of how colonization, in this case by a commensal bacteria, alters internal state and changes sensory behaviors, and in this case via modulation of sensory neuron functions via bacterially produced metabolites, and in this case such as a neurotransmitter. And so to wrap up, what I've told you about today is some of our ongoing work on understanding the mechanistic basis of how the presence or absence of specific bacterial strains, non-pathogenic strains in the gut, results in sensory behavioral plasticity in C. elegans. And in particular, we find that one major mechanism of modulation, although by no means the only one, is that signaling from the gut targets sensory neurons, directly or indirectly, to modulate these sensory behaviors. And so going forward, there's an infinite number of questions to address. As I told you, worms eat a lot of different types of bacteria. So are there other commensal or mutualistic bacteria that actually modulate other types of sensory behaviors? And if so, what are the mechanisms? What is food? What is the component of food that worms are sensing or the presence or absence of? Is it different for different types of modulation of different types of behaviors? And then whether or not sensory neurons are the targets, and that makes sense for C. elegans because it has a very shallow nervous system architecture. But it's also possible that there are other nodes in the circuit that might also be targets of gut-brain signaling, and that remains to be identified. And so in short, there is a lot of work that remains to be done. So I have had the great good fortune of working with a really wonderful group of people. So this is what the lab looks like before we all had to put masks on. So all the work on the first part of my talk was, as I mentioned, done by Asuka, who now has her own lab at Riken, and also together with Jamie and Nathan. The second part of my talk is all the work of Mike, and who's about to start his own lab, and a previous research associate, Henry Chow. The other people in my lab work on other aspects of sensory biology. I wanted to acknowledge my funding sources, our really wonderful collaborators, Bennett and Frank. And I did want to give a shout out to the really fabulous C. elegans community because they're incredibly interactive, generous, collaborative, and it's been really great to have been a part of this community for the past 3 decades or so. And thank you for listening, and I'll be happy to take any questions.
All right, thank you so much. I have a quick question. Because I'd never thought about this assay before. So in the assay where you're doing the eyelash, you quantify it by like time to turn away. Whereas when I think of C. elegans neurons and C. elegans behavior, I typically think of not dynamics, but just moving or responding in some magnitude. Have you looked at how this matches up with like internal dynamics of sensory neurons or something else?
Yeah.
Since it's specifically a dynamic measure.
Right. So the ASH neurons actually are directly connected to the backward command interneurons, and so this makes sense for a nociceptive neuron, right? So you provide it with a stimulus, it immediately elicits a backward response without actually having to go through sort of integrating interneurons in between. So Octanol responses have been looked at in ASH, and you see this nice response to it. In terms of the bacterial modulation, this has been a little more challenging to look at. So as you notice, we look at 100% Octanol responses. So 100% Octanol and 30% or dilute Octanol elicit very different behaviors. And technically, it has been very difficult to image to 100% Octanol. One of the reasons being that exposing worms to 100%, if the worms are actually directly exposed to 100% Octanol, they dissolve. So that makes things a little bit harder.
So it's hard to get data from dissolved.
Or we're trying, or Mike is trying.
Yeah.
Exactly.
Okay. Some of the questions. So Fernando Calahoro asks whether whether colonization by JU39 is impacting the pharyngeal pumping rate as well.
Yeah, Mike tried to look at that actually because so one of the things is that we thought that perhaps the food quality would also affect the pharyngeal pumping rate, which has been shown before. But I think that Mike did not find obvious changes in the pharyngeal pumping rate under these conditions. So one big difference here to notice is that a lot of the behavioral experiments that have been done with these monoamines, like anything, serotonin, dopamine, tyramine, et cetera, you put it on and sort of you look at a more sort of acute responses, right? Because you put it on the plate and you look at acute responses. The thing that's different about the growth on JU39 is that the worms are being exposed chronically. And that you can imagine could result in very different types of behaviors than the acute addition of a particular neurotransmitter. So we're trying to sort of work out the differences between the two.
Naveen Pokala asks whether OCTR-1 is sorry, I always get, there's too many letters.
GPC, protein coupled?
Whether it's a G-alpha-1, GOA1 coupled.
Do I know that? I should know that, but I don't, Naveen. I'm sorry. I don't know. I think that, I'm not going to make it up. I just don't remember.
Marion says, beautiful talk. What do you mean by JU39 is highly nutritious? Is there a memory for that?
Yeah, so there's really nice work from Buck Samuel's lab where he actually assayed a whole bunch of different types of bacteria that you find associated with the worm gut. And he looked at several different things. So he looked to see whether the bacteria support reproduction and growth. And JU39 supports wonderful reproduction, very high, very good growth. It also does not induce any of the stress reporters that typically pathogenic bacteria and other types of bacteria would induce. Mike looked at lifespan. That's also not significantly altered. And so it seems like it's not eliciting any of the starvation-dependent responses that you would expect, like a bacteria that the worms don't, it's a bad bacteria or poor bacteria in terms of nutrition. So that's what I meant by that.
Katrine Vogt asks whether starved animals reverse their behavior by just exposing them to food via sensory contact, or whether they actually have to ingest it, and whether shorter food exposure than 1 hour also reverses behavior.
Right. So Asuka actually tried this. So what she did was she exposed them. She had them starving on a plate, but she actually had bacteria on the lid of the plate. So they were being exposed to the odors that were made by bacteria. And that was not sufficient to actually make them act like they were fed. And so it seemed like they actually have to eat the bacteria. But you can also feed them antibiotic-killed bacteria, which would be producing some metabolites, but potentially not all the volatile odors. That was completely sufficient to also mimic the fed condition. And in terms of the short time frame, so for the thermotaxis behaviors, it really takes 2 hours for the disruption to manifest itself. And there is literature in flies and also in worms where different periods of starvation. So there is short, like fasting, and then there's starvation, prolonged food deprivation. And on different time scales, you engage different neuromodulatory mechanisms and engage sort of different components of the circuit, because there's a difference between saying that there is sort of transient food deprivation as opposed to chronic food deprivation. And so it's not surprising that actually there'd be differences between how long you're actually depriving them of food. And this is kind of an interesting thing that we've also started to think about.
Just to follow up on that, do you know if the differences are because of the time to produce the insulins or whatever, like just to start that production, or like to release that amount?
Yeah, no, that's a good question. So our idea is that perhaps, we, as I showed in the thermotaxis case, it requires DAF16, and so presumably there's changes in gene expression. But you don't really need 3 hours for that, right? So there's probably some cascade of events that has to occur in order for the behavioral disruption that we see. And we don't actually know what that is. So people have done these food deprivation experiments where you see massive changes in insulin, all the various insulin genes, massive changes in the expression all over the place. But typically they're done for, an hour or so at most, and not for that much longer.
Ethan Yu is asking how long the worms need to be cultured in JU39 to show a food preference?
Right. So it has to be, so all the experiments that I showed you were done with the worms cultivated the entire, for 1 generation. But I think Mike has done these experiments where he can shift them from OP50 to JU39 in the L4, the 4th larval stage. And that's sufficient to also allow the modulation that we see. One thing to note is that a lot of the colonization that we observe appears to happen in the adult and not on the L1 larvae. And so I don't know why. And so that sort of affects the way we do these experiments.
Yeah. Nipun Bizrar, says, really fascinating. Do you know where in the worm the tyramine is converted to octopamine?
Really good question. Yeah, so Mike has been trying to address this. So the octopaminergic enzyme and the tyraminergic enzyme typically are actually overlap. Mm-hmm. But there are some cells and tissues where the TDC1, the one that makes tyramine, is made, where octopamine is not made. So if bacteria in the gut are making tyramine, somehow it's got to get to those tissues, right? So there's a few neurons, there's also the gonad sheath. And we actually currently have no idea where this is actually happening or how. So we don't know whether it's just diffusing, whether there is some kind of monoamine transporters that actually got to take it into a particular neurons. Mike is definitely trying to address this question.
Nick Burton says, great talk. Is there a particular reason you experiment with JU39 and Octanol, or do you think these types of effects might be quite common and many bacteria would do the same thing?
Yeah, so Mike actually tried a whole bunch of different bacteria. It's actually in the paper, and none of them affected Octanol in any way. But I think that at this point, any bacteria that makes tyramine probably would also have the same kind of an effect. He kind of started working with this bacteria in Octanol because he found an effect, and so we wanted to work out the mechanism. But he in his own lab is really interested in sort of expanding this to other bacteria that the worms eat, and just also expanding the behavioral output, because he's essentially looked at a tiny number of chemicals, right? And worms, as you know, have a large number of behavioral responses, and maybe a lot of them are modulated by bacteria. We just don't know. Mm-hmm. It's just sort of the tip of the iceberg. He's going to do fabulous work in his own lab.
Abhilasha asks, isn't heat shock response responsible for thermoregulation in worms, which is generally found in eukaryotes?
So the heat shock response is, so interestingly in worms, at 25 degrees, you actually do see a heat shock response as well. And there's been work showing that the heat shock response may actually modulate some aspects of behavior. But the behaviors that we are looking at, the very sensitive temperature change that they have to navigate when they navigate thermal gradient, do not appear to be dependent upon the heat shock response. So there are other aspects of behavior that are.
Fernando asks now, what is upstream of the insulin released by the intestine? And if OP50 doesn't colonize the intestine, what is the cue for the insulin release?
Right, great question. So we thought that there was going to be regulation of insulin transcription. There does not appear to be, so it might be insulin release. There's also actually evidence of insulin to insulin signaling. This is work done from Yun Zhang's lab, so there could be something else in between. So what is regulating the gene expression? No idea. There is activity-dependent gene expression, CREB, potentially. So there's a whole bunch of different transcription factors and other molecules that we have candidates that we're going to look through. But right now, I actually have no idea at all. And if it's actually released, which is also is a different way of modulating the downstream activity, that's also something that we don't know anything about.
So I'm going to ask you a really speculative, broad question. So answer however you want. So I've always been really fascinated by, in C. elegans, the fact that these different sensory neurons seem to, maybe they're somewhat well-defined in terms of AFD being a thermosensor and, I'm blanking on the neuron that's the aversive one.
ASH?
ASH, yes, thank you. And then the others have maybe some other variety of behaviors, but it really seems like they coordinate a lot in terms to refine the behavior. I'm just really curious whether, thinking your first talk, how you think about these sensory neurons working together and whether you think AWC and ASH and, ASI and whatever have special behaviors or like ways of sculpting the behavior, or whether it's really the coordination of each of them kind of together makes the behavior, if that makes sense, whether it's like a distributed or just how you think about this, because it's something I've never had like a good way to think about. Right.
So one of the things about the worm sensory neurons is potentially possibly because they have to respond to so many different cues with such a small number of neurons. Each neuron, I just don't think that there's any neuron that's unimodal. They're all polymodal, right? So they're responding a lot of different orders. But there's a lot of coordination. So if you look really carefully, there's pretty much. So if you look at there was a really beautiful paper from Corey Bargman's lab actually, where they showed, so there's a neuron called AWA that responds to this chemical called diacetyl. This is butter odor. But they actually found that if you put the worm, expose them to diacetyl, AWA shows a great response, and the worms love diacetyl, they go towards it. But if you optogenetically activate AWA, you don't see the same type of response. And so what they ended up finding was that addition of diacetyl actually not just activates AWA, but it actually also activates other neurons, and all of this coordination results in sharpening the response to diacetyl sensed by AWA. And so there's an incredible amount of coordination among all of these. And as you yourself know from being a graduate student in Shrek's lab, that there is also a lot of crosstalk between sensory neurons at the sensory neuron level, right? Mm-hmm. So sensory neurons can themselves also act as interneurons in the context of neuropeptidergic signaling. So I think there's an enormous amount of coordination among all of these neurons. And one of the interesting questions is, where does this sort of modulation occur? Because interestingly, it's still not clear where all the receptors are and where the release happens. And so these are sort of questions that I think the field has to address.
I think the last question is from Ann Nguyen, who's asking, do you know anything about how tyramine is synthesized by JU39 in the gut?
Yeah, so it's by those enzymes that it has. So it has got these two enzymes that are encoded by the genome. Oh, you might be asking about why it's made in the gut. So the operon is actually induced by both tyrosine. So when the bacteria takes up tyrosine, it induces that operon. The other thing that also induces the operon is low pH. And so the idea is that one of the reasons we don't see tyramine when you just have the bacteria on its own is because if you lower the pH, you can see induction. So once it gets into the gut, which I think has I'm going to make this up. The warm gut has a pH of about 4-ish, I think, but that's sufficient to actually induce that operon and cause tyramine production. And in fact, tyramine production by bacteria typically allows it to survive low pH environments. That's one of the reasons that they make it.
Cool. Thank you so much for speaking.
Thank you, Adam.
And thanks everyone for being here. And this is the last one for the season, so we'll start back up in sometime in September. So thanks everyone.
Great. Thank you. All right, everyone. Thank you. Have a great summer.